The universal IgG lateral flow assay is a highly sensitive, research-use-only device intended to detect human IgG antibodies specific for any given antigen. The idea is to use this platform to quickly verify if a new antigen holds promise for a serological test. Here we will illustrate the power of this test with an application to COVID-19. We have selected one of the coronavirus proteins and have biotinylated it, and we want to know if we could build a test using this biotinylated antigen. As for our samples, they contain several concentrations of human antibodies that should be specific for the antigen we have selected. The serum sample is first mixed with a biotinylated antigen. Here we have selected an antigen, the RBD domain of the coronavirus. The mixture is added to the round port of the test. The chase buffer is added to the square port. The sample is then read at the 30-minute mark. This system is highly sensitive. We can see a nice dose-response curve. The test detects a 1 to 4,000 dilution of the positive control, which is an excellent performance. In this manner, we have shown very quickly that the biotinylated antigen is adapted to detecting antibodies to SARS-CoV-2. The next step is for us to dry that antigen in the test strip so that we can have a finished assay.